It is very important in this section of the course that you understand and be comfortable with working with Punnett squares. Punnett squares are used to determine what kind of potential offspring that you could have from crossing two parents. You can get an idea of what the both genotype and phenotype makeup is of both the parents and the offspring. Genotype refers to the genetic makeup, specifically what alleles the organisms possess, whereas phenotype gives you an idea of what those traits would look like, or the overall appearance of those organisms. What allows us to be able to determine what gametes would be produced from a certain organism, we rely on that law of segregation that states that these alleles will separate out into separate gametes. This allows us to construct our Punnett square, or construct our table, with the gametes on the top and the left side from each parent. The Punnett square, in essence, simulates fertilization, so it will take a look at what kind of gametes can be produced by individuals and allow you to determine what would happen if those gametes were fertilized with each other. At the end, we can count our individuals and calculate our offspring ratios. We can see here in this diagram to the right, we have a heterozygous male. Let's consider that we have a heterozygous male as well as a heterozygous female. As a result, when we have a heterozygous individual, we know that a heterozygous individual carries both the dominant and recessive allele. So we know when we state something as heterozygous, we know what the genotype of that individual would be. In this case, we have a big R, small r genotype for our parents. As a result, the male in this instance, half of the gametes would have a big R, half of them would have a small r. This is what we see at the top of the Punnett square. Let's also consider that we have a heterozygous female, and it would also produce the same gametes. When you simulate fertilization and try and determine what kind of offspring you would have from these two individuals, this is what we see in the middle of the Punnett square. So if we had a big R from the male that joined and fused with a big R from the female, we would have a big R offspring. If we had a little r from the male fused with a big R from the female, we would have a heterozygous individual. If we take a look at the bottom left of the Punnett square, this individual results from a big R from the male and a small r from the female. And then finally, if we have a small r from the male joining with a small r of the female, we would then have our recessive individual. We can glean a lot of information from the Punnett square. When we take a look at our offspring ratios, we can come up with a genotype ratio such that 1 out of 4 of these individuals would be big R, or homozygous dominant. Another quarter, or 1 out of 4 of our offspring, or 25% of our offspring, would be homozygous recessive, small r. And 50%, or 2/4 of our offspring, would be heterozygous, big R, little r. Keeping in mind that half of these heterozygous individuals would have gotten the dominant allele from the dad, and half would have gotten the dominant allele from the mom. We can also get an idea of what our phenotype ratio would be, looking at what these offsprings would look like. And in this case, we have 75% of the offspring would be round, and 25% would be wrinkled. It is important to understand and be able to state clearly what a phenotype and genotype ratio are. When we are considering our cross, say similarly to what Mendel did, taking the F1 hybrid generation and crossing it or self-pollinating those individuals, when we would get our 3-to-1 ratio. That 3-to-1 ratio refers to the phenotype ratio, such that 3 of those individuals were purple and 1 of those individuals was white. If we take a look at the genotype breakdown, however, that breakdown gets a little bit more complicated because the genotype ratio is a bit more specific. We have 1 individual that is homozygous dominant. We also have 1 individual that is homozygous recessive. And then we have 2 individuals that are considered heterozygous. So this means that our genotype ratio would be 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. When you are looking at your ratios and stating them, it is also important to be sure that you're clear in what you state, meaning the genotype ratio being not simply 1 to 2 to 1 as we see on the bottom of this diagram. When you're stating a ratio, you should clearly indicate that you have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Similarly, when stating a phenotype ratio, you should also be very clear and state that you have 3 purple individuals to 1 white individual, as opposed to simply saying that you have a phenotype ratio of 3 to 1. We're now going to do another example of a monohybrid cross. A monohybrid cross looks at simply one trait. We will see when we get to the further slides that we can have dihybrid crosses that look at 2 different characters. So here we've got a pure breeding green plant that is crossed with a pure breeding yellow pod pea plant. So we can get an idea of what our parental generation looks like. Here we have a green pea pod plant crossed with a yellow. We are also given a little bit more information. We're told that all of our offspring are green. This is an important point because this allows us to determine what is dominant versus what is recessive, and this also allows us to determine what the parental genotype and also the F1 genotype would be. So this means that if all of the offspring are green, the yellow trait must be recessive. So now we can assign the alleles. Here we know that green would be considered dominant, so we can refer to it as being a big G, whereas yellow would be a little or recessive allele, little g. Now we know, going back to the beginning of our question, that we are dealing with pure breeding plants. As a result, we know that our green plant would be big G, and our yellow plant would be little g. We also know that the gametes from those individuals must have been big G from the homozygous dominant parent and little g from the homozygous recessive. Now, we could also know that the offspring genotype for our F1 generation would have been big G, as they all would have been hybrids. So now we self-pollinate, or we cross two different offsprings from our F1 generation. If our F1 generation are all hybrids, that is, in essence, we are taking a heterozygous individual and crossing it with another heterozygous individual. Now we will make our Punnett square to determine what our F2 generation would be. We first have to assign alleles. So if we have a heterozygous individual, big G, little g, we now have to consider what kind of gametes would this individual produce. If I had a hybrid individual as such, it would produce half of the gametes with a big G, and the other half of the gametes would be a small g. Similarly, for the other parent, we would have the same kind of gametes produced. Half of them would be a big G and half would be a small g. Now we can complete our Punnett square, and we can see the offspring results here. We can also calculate various ratios. If we wanted to consider our phenotype ratio, we would have 3 green individuals to 1 yellow, the green individuals being those with the genotype big G, or big G, little g. We could also state that phenotype as 75% green to 25% yellow. That would be acceptable as well. If we also take a look at our genotype ratio, we would see that we have 1 homozygous dominant, 1 big G, to 2 heterozygous individuals, big G, little g, to 1 homozygous recessive, little g. Another important concept to understand is that of a test cross. Test cross can be very helpful to figure out the genotype of a parent. Specifically, are they homozygous or are they heterozygous? It is important to remember that when you are doing a test cross by definition involves a homozygous recessive parent. This means that when you're crossing a homozygous dominant with a recessive, that would be considered a test cross, or if you're crossing a heterozygous with a recessive. By base definition, a test cross has to have a homozygous recessive parent. So this is important in being able to figure out genotypes, because if you have a homozygous dominant crossed with a recessive, you would get all hybrid individuals as your offspring. However, if you had a heterozygous individual crossed with a homozygous recessive, you would then see the recessive trait in the offspring. So basically, by crossing a parent with a homozygous recessive, if you can tease out that recessive trait, the only way you can do so is if that other unknown parent is heterozygous. If that trait is fully masked, then you know that the other unknown parent must have been homozygous dominant. This slide here depicts the test cross as described in the slide previous. We have a dominant phenotype purple plant, however, we do not know what the genotype is. That individual could be big P, or it could be big P, little p. We will do a test cross and cross it with a fully homozygous recessive. And with that parent, we know what the genotype would be. It would be little p, little p. We have two scenarios to consider. The first scenario is if we have a homozygous dominant individual, and this is outlined in the left of this diagram under predictions. We can see that if we had a fully homozygous dominant individual, the only gametes that could be produced by that individual are big P, big P. We can also see that in our recessive individual, the only gametes that could be produced by that parent would be little p, little p. As a result, when you undergo fertilization and you carry out your Punnett square, you would see that all of the individuals are considered to be heterozygous, big P, little p, and all of them would exhibit that dominant phenotype, so all of the offspring would be purple. In the second scenario, we would consider the unknown parent to be heterozygous, big P, little p. If that were true, then the gametes that would be produced by that individual would be half of them having the dominant allele, big P, and the other half would have the small allele. Similarly, we are doing a test cross, so the gametes produced by the other parent would simply be little p, because we're crossing it with a homozygous recessive individual, and those are the only gametes that that individual can produce. In this instance, we can see that the genotype and phenotype ratios of the offspring differ. We would have 50% of the individuals being heterozygous, big P, little p, and exhibiting the dominant phenotype being purple flowers. And the other 50% of the offspring would be little p, exhibiting the recessive phenotype or having white flowers. So we can see in our results that when we have a fully dominant parent as the unknown genotype, all of the offspring are purple, meaning that even though we're crossing it with a recessive, that is going to be masked because only the dominant trait will be exhibited. However, if we have a heterozygous individual as our unknown genotype, and we cross it with a recessive, we would see the recessive phenotype in the offspring ratio because it would not be masked, and one of the small alleles from the heterozygous parent would be given to the offspring. Mendel and his experiments also led to the development of the law of independent assortment, and this was discussed when we went through the slides on meiosis. The law of independent assortment states that when gametes are formed, these alleles will segregate independently of other alleles. That means that all the traits from the parents are inherited independently. This is important when considering the tetrads. Remember, in meiosis 1, we have the formation of the tetrads and the tetrads lining up on the metaphase plate. The law of independent segregation has to deal with when those tetrads are pulled apart. It's random which one goes to either side, but you have the separation of those alleles, the mother and the father alleles. When we're dealing with the law of independent assortment, remember, this is how those tetrads separate out when you have multiple tetrads lined up on the plate. So the law of segregation describes how those alleles separate within the tetrad. Law of independent assortment states that when you have the first tetrad separating out, it has no influence on how the second one separates out. Meaning that if your dad's original N copies were pulled to the left in chromosome 1 and your mom's were pulled to the right, that has no influence on how chromosome or tetrad 2 is separated out. Meaning that it's a 50-50 chance of your dad and mom going to either side. Both the law of independent assortment and the law of segregation take place in anaphase 1 of meiosis. However, they deal with two separate concepts. Because we have the law of independent assortment, this allows us to determine what the gametes would look like when we're dealing with more than one trait. So we are now going to take a look at what happens in dihybrid crosses, looking at two different traits. We can predict what kind of alleles that we would have from our individuals based on the law of independent assortment. Let's take a look at a typical dihybrid cross. In this instance, we are going to be taking a tall purple plant and crossing it with a dwarf white plant. Keep in mind that in a dihybrid cross, we are now dealing with two different characteristics. In this case, tall versus dwarf and purple versus white. Let's say that you are given that both of these individuals are considered to be homozygous. As a result, we have a homozygous dominant individual that is crossed with a homozygous recessive. If we consider what kind of gametes these individuals can produce, the tall purple homozygous dominant plant would only produce gametes that have a big T and a big P. Similarly, the homozygous recessive individual would only produce gametes that have a little t and a little p. If you had fusion of these gametes, you would get your F1 individuals, and all of those would be heterozygous. Their phenotype would be tall and purple, because tall and purple are the dominant traits. If we now cross individuals from our F1 generation to figure out our F2 generation, we also need to consider what kind of gametes could be produced by these individuals. Because we have the law of independent assortment, that means that these would sort independently of each other, trait 1 versus trait 2. This means that we have 4 potential gametes that could result. Big T, big P, big T, little p, big P, and little t, little p. Similarly, if our other parent was male, it would produce the same types of gametes. So now let's do our Punnett square. We line up one parent's gametes on the left side, and we line up the other parents, in this case the sperm gametes, on the top. We can now determine what our offspring potentials are. If we had big T, big P egg fusing with big T, big P sperm, we would then have big T, big T, big P as our offspring. If we continue along that row, we would determine the rest of the gametes, resulting from a big T, big P egg and our corresponding sperm on the top. We would consider this for every single egg potential gamete that we have, and these are the resultant offsprings. Keep in mind when you're doing a dihybrid cross, it is important to keep your like alleles together. You can see that the T's are kept together and the P's are kept together. This is the correct format for entering in your offspring ratios. If we now consider what the phenotypes would be, here in this instance we have a tall and purple plant. We also have tall and purple. Anytime that you have a dominant allele, it would exhibit that dominant trait. And continue on. Here we have a tall but white flowered plant, another tall and white flowered, so on and so forth. We only have a dwarf plant when we have little t, and we only have a white flowered plant when we have little p. So we can see the offspring ratio here. If we take a look at the overall phenotype ratio, we would see that it is a 9 to 3 to 3 to 1 ratio, meaning that we have 9 fully dominant individuals. So 9 would be purple and tall. 3 would be purple and short. Another 3 would be tall and white. And only one individual would exhibit the fully recessive state, meaning that they would be dwarf and white. So in general, when you are crossing two heterozygous individuals for two traits, this will yield a phenotype ratio of 9 to 3 to 3 to 1. However, it is important to understand where that ratio comes from. You could also be asked to give the genotype ratio of this type of cross, which would be very complicated. However, it is still doable. You would take each individual genotype and count up which ones were similar. In this case, you would start with big T, big T, big P, big P, and you would note that there is only one offspring in the offspring potential Punnett square that has that specific genotype. And you would do that for all of the other genotypes below.